The overall goal of this protocol is to predict drug binding to Caseum from tuberculous lesions.
This method can help determine how effectively antimicrobials penetrate the caseous core of necrotic lesions without the use of animal models and expensive imaging techniques. Although this method can provide insight into tuberculosis drug discovery, it can also be applied to other diseases that are characterized by the formation of lesions or abscesses where drug penetration is limited. Visual demonstration of this method is critical, as consistency in the performance of the protocol will ensure reproducibility of data between experiments. Today, I will demonstrate the cell culture procedure and preparation of the surrogate matrix. Hsin-pin, a research associate from our lab, will demonstrate the red assay.
Grow THP1 monocytes in T175 cell culture flasks containing 80 milliliters of rPMI 1640 medium with 2 millimolar of L-glutamine and 10% fetal bovine serum. Incubate the flasks at 37 degrees Celsius with 5% carbon dioxide for 3 to 4 days. Once the cells are about 70% confluent, split them into two 50 milliliter conical tubes and centrifuge them at 150 times G for 5 minutes. After the spin, discard the supernatant and suspend the pellet in 10 milliliters of rPMI 1640 medium. Pipette 5 microliters of this culture into a 1.5 milliliter tube containing 45 microliters of trypan blue. Mix thoroughly by pipetting. Transfer 10 microliters